Does anybody, so this is where we left off. This is the last slide I talked about last time. Does anybody have any questions about this idea here? The notion that 2,3-bisphosphoglycerate is stabilizing the T state, and shifting hemoglobin towards that T conformation, the affinity for oxygen typified by that. Are there any other questions about anything? Any other material? Speak now if you have. Nope. Okay. Well, let's look a little bit more closely at 2,3-bisphosphoglycerate. It is a glycolytic intermediate that is produced by respiring cells, specifically respiring cells that are at or beyond the anaerobic threshold. So in general, who has not had glycolysis, central metabolism before in a class? So everybody here has had a little bit of glycolysis before? Okay, good. You'll remember then from glycolysis, it's the basic process by which the cell breaks carbon-carbon bonds and takes that energy that's released and produces ATP with it. These carbon-carbon bonds are in glucose, and we break glucose down. There are several steps by which we get to 1,3-bisphosphoglycerate. Then typically, glycolysis proceeds through phosphoglycerate kinase. It's okay, I'm going slow through details that aren't that important, just to wait for everybody. Come on in, sit on that. Phosphoglycerate kinase takes 1,3-bisphosphoglycerate and transfers one of the phosphate groups from 1,3-BPG to ADP and produces a unit of ATP, leaving us with 3-phosphoglycerate. However, there is a suite of other enzymes here. Bisphosphoglycerate mutase and 2,3-bisphosphoglycerate phosphatase. Two fun-sounding enzymes, which are affected by the pH in the cell. So if the cell has a low pH, that is a high hydrogen ion concentration, then bisphosphoglycerate mutase is upregulated, or I should say potentiated. It's potentiated. It becomes more active. And this enzyme, the 2,3-bisphosphoglycerate phosphatase, is inhibited. And so what happens here is we start shunting 1,3-BPG instead of directly to 3-phosphoglycerate. We start shunting it over here to 2,3-bisphosphoglycerate because of the mutase group. The mutase functionality takes the phosphatase, or the phosphate group that is on the 1 attached to carbon 1 and shifts it over to carbon 2. This molecule here is the effector that we're talking about in the previous slide with hemoglobin. All right. So 2,3-BPG is a signal that the cell is perhaps producing, so the pH is going down. This means that carbon dioxide levels or carbonic acid must be going up. It means it's respiring rapidly. Perhaps even lactic acid is being produced if we're in anaerobic metabolism primarily. So the pH is going down, the hydrogen ion concentration is going up. And it's a signal that the cell is rapidly respiring if 2,3-BPG starts showing up. So here's the binding pocket where you'll see 2,3-BPG in hemoglobin. All right, and it fits itself right in the donut hole. It's kind of like it's like the what are those things called in Dunkin' Donuts? The donut holes, right? The little, what was that? Munchkins. It's like the munchkin. It fits. Thank you. It's been a while since I had a munchkin. It fits itself right in the center of the hemoglobin. And you can see here how there are these 6 different residues, these 6 different amino acids, 3 on each of the 2 beta chains that coordinate 2,3-BPG, which is what makes the binding pocket. So we have a histidine, a lysine, and another histidine. One set on each of the beta subunits of hemoglobin. It fits right in there and prevents that conformational shift, that 15-degree shift of orientation of one set of the subunits with respect to the other along the axis of symmetry in hemoglobin. Right, just fits in there and kind of it's like a little bit of a rock in the door, preventing the door from going, or something like that. A wrench in the works. But you can see how it may, in fact, stabilize the tense conformation. It's limiting the conformational mobility that hemoglobin can have, because there's those subunits can't collapse down on them on each other because there's this thing in the way, this molecule in the way. Following this? Yeah. Okay. So what's babies do not have the same hemoglobin as you and I. And I shouldn't say babies. Fetuses do not. In utero fetuses, babies in the stomach, unborn fetuses, they have a different hemoglobin. It's called fetal hemoglobin. And their beta subunit is replaced by a gamma subunit that has the following mutation. The histidine 143 in the sequence is replaced by a serine. And because of that, 2,3-BPG has a reduced affinity for fetal hemoglobin. The 2,3-BPG does not fit in the binding pocket. The munchkin doesn't fit in the hemoglobin quite as well. All right? So what effect do you think this is going to have? Can anybody guess? Yeah. Yeah, this T state will not be as stabilized, right? So the R state is going to be a little bit more favored than in the maternal hemoglobin, isn't it? So what effect is that going to have on the binding curve? You're totally right. Is that going to shift the curve to the right or to the left? Do you guys remember what that? To the left. To the left. I'm juggling my brain. Yes, to the left. So let's take a look at that. In blue is maternal red blood cells, maternal hemoglobin, and with alpha and beta subunits. And in the red, we have the fetal hemoglobin with this histidine to serine substitution and a reduced affinity for 2,3-BPG. Now, if we look at the same partial pressure of oxygen at some place in the periphery, specifically the placenta, where the baby is attached to the mother and draws its nutrients and oxygen from her, we'll see that the maternal, so we'll say that concentration is down here, for example. At this concentration, oxygen is going to have a much higher saturation level, a much higher affinity for the baby's hemoglobin than it will from the mother's. This is how baby fetuses are able to draw oxygen off of the mother's hemoglobin. All right, the cards are understandably stacked in favor of the baby's hemoglobin. Right, and this is how we get a hemoglobin transfer to, oxygen transfer to the fetus, and then blood passes through the fetal circulatory system and is delivered to the peripheral tissues of the infant. All right, is this all good so far? Well, you know what? I should have put another slide in here because it's so interesting. I'm going to take a little bit of a diversion just to tell the story a bit. Should have a better picture here for you guys to look at. But so remember when we were talking about the heart, and I talked about how blood flows from the right atrium to the left atrium through the hole in the heart that's in the atrial septum in the wall called the foramen ovale in the fetus. Right, so that's because blood that's coming from the placenta through the umbilical cord into the fetus comes directly into the portal system of the liver. It goes first to your liver, and the baby's liver gets first dibs, so to speak, on any sugar, glucose, that it is able, or other nutrients that it's able to glean from the mother's blood. So that the liver stores that as glycogen and is able to keep, which is how the baby keeps its blood sugar level. From the liver, that blood is now on the venous side. It's on the venous side. It's low pressure blood. It's going to go back to the right side of the atrium. Before we send that blood to the lungs, it doesn't need to go to the lungs. It's already oxygenated, isn't it? It picked up the oxygen right here from the mother's hemoglobin. So rather than send it to the lungs, we want to just send it right to the tissues. And so one way is through the foramen ovale, and the other way is through something called the ductus arteriosus. I think I pointed this out as well. The ductus arteriosus is a little shunt that connects the pulmonary trunk that's coming out of the right ventricle it connects that pulmonary trunk to the aorta, and blood is able to pass directly from the right side of the heart to the systemic left side without having to go to the lungs first. Some of it, of course, goes to the lungs, but a lot of it is shunted over to the left side of the heart through the foramen ovale and the ductus arteriosus. Upon birth, immediately postpartum, what happens? What's the stereotypical thing? What's the kind of caricature of babies being born? Baby comes out, and then what happens? It cries. It cries, and it used to be a kind of dubious task of the obstetrician to smack the baby on the backside to get it crying, right? The reason for this is if that baby takes a deep, full breath, if it's going to start squalling, then it's going to fully inflate those lungs, and you want to have that is going to spike the blood pressure on the pulmonary side of the cardiovascular system on the pulmonary side of it. That'll spike the pulmonary blood pressure, and in so doing, immediately slam shut that foramen ovale. There's a little flap there that slams shut that first breath the baby takes, and it also causes smooth muscle fibers in the walls of the ductus arteriosus to clamp down really hard, shutting off that. So within that first breath, it is truly a miraculous moment when the infant's entire cardiovascular system completely switches over to an entirely new flow pattern than it was having for the time that it was in the mother's stomach, or in the womb. It's not in the stomach. So as soon as this happens, the baby goes through another long process, long meaning several day process, up to a week or so process of switching over its hemoglobin from fetal hemoglobin to the hemoglobin postpartum, the hemoglobin that we all have. Yeah, maybe I'll get to the rest of the story when I talk about hemoglobin turnover, or maybe I can just tell it now. As hemoglobin is processed, the way the body processes it, the recycling of hemoglobin, the body rips the porphyrin ring, the heme molecule, out of the hemoglobin. The protein itself gets digested and recycled, and then the porphyrin ring, we have the iron gets extracted, and then the porphyrin ring itself goes on to help produce some of these pigments called bilirubin and biliverdin in the liver. And these are chromophores that, there are chromophores here. This is why our blood is red, of course. But when the iron has been removed, they're still chromophores because of all those pi electron networks, those double bonded conjugated networks that you see in the porphyrin rings. They are the origin, I'll say it this way, of what's called jaundice. So people who have some sort of liver dysfunction have a high turnover there's a problem with the turnover and the production of bilirubin and biliverdin, and that leads to a yellowish tinge in the skin. Some newborn babies can have that same yellowy jaundice color due to the rapid turnover of the fetal hemoglobin. They'll be all right. Just put them in the sunshine a little bit, and they'll turn over. Sometimes you can. There are other interventions for that, but it's not that big a deal. All right, I won't divert any longer. But the important part here is simply that fetal red cells have a reduced affinity for 2,3-BPG, which shifts it slightly towards the R state and allows it to take oxygen more readily from the maternal hemoglobin. Okay? The other stuff about the circulatory, that was gravy. Physiological story. So, 2,3-BPG is not the only effector of hemoglobin saturation dynamics. There's a surprisingly large list of things that can affect hemoglobin function. And in fact, the list is far longer than we're going to be able to go through. But I'm going to go through a couple of the interesting ones. All of the effects on hemoglobin make sense if you think about them from like a common sense physiological standpoint. All right, so you can go about memorizing all of these if you want, or you can just think about the effect of temperature. What would you want? How would you want hemoglobin to respond to temperature or to pH, etc.? All right. So let's look at these. Let's think about. We'll do pH first. pH is what's called the Bohr effect, and this is not Niels Bohr. This is named after Christian Bohr, who was Niels Bohr's uncle or something like that, related to that guy. But he was the first person to recognize the effect on the efficiency of the body's ability to transport oxygen as it relates to pH. So as the blood becomes more acidic, the curve shifts towards the T state, and as it becomes more alkaline, it shifts towards the R state. This should make sense to you. So the closer the hemoglobin is to the T state, the more the hemoglobin is going to want to offload oxygen. All right, it's going to want to offload oxygen in the T state. So let's talk about a particular partial pressure of oxygen. Normal would be here at purple, and say you're respiring really rapidly, you're driving your pH down in your cells, you're producing lots of carbon dioxide, which is forming carbonic acid, you're forming lactic acid, pH is going down in the peripheral tissue. You're going to want to offload more oxygen. You want to tune the hemoglobin to be able to unload more oxygen. So that relationship should actually make sense to you, all right? Do you guys follow the logic of that? So rather than having to memorize the relationship, you can just think about what makes physiological sense, because that's what physiology is. It makes sense. It should. Sometimes it doesn't. Mostly it does. And then temperature. Similarly, all right, as temperature increases, we also move towards the T state. Meaning as temperature increases, we're also going to want to offload oxygen. Hemoglobin is going to be more likely to offload oxygen. Well, does this make sense? What's the main way your body raises its temperature? Being active, right? Moving, using your muscles. That requires internal respiration of the peripheral tissues. So as the increased demand for oxygen goes up with temperature in those cells, the hemoglobin is at any given partial pressure of oxygen, as the temperature increases, it's going to be more inclined to unload oxygen. All right? And in fact, the effect is really extreme, you can see. So what do I have? What are these bullet points? Deoxyhemoglobin has greater affinity for H plus than does oxyhemoglobin. Yeah, it's just saying the same thing. But the way to think about it is in terms of physiological common sense. All right. So another effector is carbon dioxide. So the red line is the pH, is the saturation curve for hemoglobin at physiological pH with no carbon dioxide present. Now, the blue line is at a lower pH. We're shifting the curve. This is the Bohr effect. Still no carbon dioxide present. And then the last line is pH 7.2, so acidic conditions at 40 torr of carbon dioxide. And it's shifting the curve even further. So this should also make sense. As carbon dioxide concentration goes up, the more carbon dioxide, the more the hemoglobin is going to favor the T state and want to unload oxygen. All right, it's going to want to unload oxygen because it's at the peripheral tissues. As carbon dioxide concentration is going up at the peripheral tissues, that's a signal for the hemoglobin to unload its oxygen because it's where it wants to be. And the more carbon dioxide, the higher the oxygen demand at the tissues. Y'all with me? All right. Friday. We're almost there, folks. So the last effector I'm going to talk about, I don't know, I don't like to put students on the spot too much, but I truly hope there are no smokers in this classroom, is carbon monoxide. Carbon monoxide. So carbon monoxide it has a binding constant that's 200 times, over 2 orders of magnitude, larger than that of oxygen for hemoglobin. Carbon monoxide likes to bind to Fe2 plus and stay there. It stays there. And as you increase the carbon monoxide poison, so the downside of this is that once you get carbon monoxide on that hemoglobin, that is taking that hemoglobin out of the effective population. All right, it's taking it out of the functional population of hemoglobin. It can go back and forth from the lungs to the tissues, and back and forth 200 times more than a hemoglobin that has oxygen bound to it, and that carbon dioxide is still going to be there. All right, so this means that exposure to even low levels, chronically low levels of carbon monoxide can be very dangerous. This is what carbon monoxide poisoning is. And it's real, folks. Back in my wild and woolly youth, when I was your age, I was in a band living in Ann Arbor, and there was this great guy named Jeff that was the local recording studio guy in Ann Arbor that recorded all the bands. Lived in kind of a cheap trailer was his home, and he put all his money into his recording studio. Great guy, helped everybody, and he died because a furnace in his house was not properly ventilated, didn't have the proper air return for combustion, and his trailer one night filled with carbon monoxide, and both he and his mother died. It's real. So I have a picture here showing how times have changed happily. There's a lot of differences between this science classroom and the one depicted here. The first is that, happily there's a lot more females in this classroom than that one. But all these guys are sitting here smoking. Teachers holding forth with a cigarette. All those guys are in there discussing whatever they're discussing with cigarettes in their hand. Apparently this was common practice back in the day. I used to teach in Texas for a while, down there, Texas A&M, Corpus Christi, and I walk into my anatomy and physiology classes to teach, and I have to walk through this cloud of smoke at the outside of the building as all the students were smoking super hardcore before the exams. I was always thinking that is the worst possible thing you can do before an exam. Put carbon monoxide on all your hemoglobin and reduce your ability to think clearly on the test. So if you are going to smoke, please wait until after the test. Do yourself a favor. Don't smoke. Okay. Carbon monoxide. Any questions on that one? No? Okay. All right, so here's kind of a summary slide of all this. So things that decrease the partial pressure of 50% saturation. That's what this P50 means. P50 is the partial pressure of 50% saturation. Things that decrease it, meaning you'll have an increased affinity of hemoglobin for oxygen. So meaning at a lower partial pressure, you'll reach 50%. Things that increase the affinity are a decrease in temperature, a decrease in the partial pressure of carbon dioxide, a decrease in 2,3 BPG, bisphosphoglycerate, diphosphoglycerate, DPG, same molecule, or an increase in pH. And then conversely, an increased partial pressure of 50% saturation or a decreased affinity is correlated with increase in temperature. That should make sense. As the body temperature goes up, you want to deliver more oxygen to the tissue. An increase in carbon dioxide partial pressure, again, a signal of respiration in the cells. An increase in 2,3 DPG, that's kind of a proxy for pH, and also a signal of active respiration in the cells, so wanting to unload more oxygen. And then, of course, the decrease in pH, which correlates with carbon dioxide concentration, lactic acid concentration, and is really a signal of increased respiration and oxygen demand. Consequently, a greater predilection to unload oxygen or decreased affinity for oxygen. You guys follow all those connections? Just blew through a lot of stuff. Had a few tangents along the way. Yeah? Okay. So let's summarize gas transport, and as we do so, I'm going to introduce the idea of what to do with all this carbon dioxide. So I've talked about how to get oxygen from the lungs to the cells, and the different things that affect the unloading of oxygen at the cells, the different effectors on hemoglobin. And then I'm going to, at the end of this slide here, talk a little bit about carbon dioxide transport. So first of all, there's diffusion across the 0.5 micron membrane in the alveolar airspace, where oxygen is picked up, mostly, the vast majority of oxygen is picked up by hemoglobin. There is a teeny tiny amount of oxygen that gets directly dissolved in the blood. Does anyone here have their scuba certification? Yeah, so you guys are familiar with that, right? The bends, if you come out, you go under pressure, you go to depth, and if you come out too rapidly, the different blood gases, oxygen and carbon dioxide, that are dissolved in the blood directly, not the ones that are necessarily attached to hemoglobin, but the ones that are directly dissolved, they can come out and you can form bubbles in the blood, causing the bends. Dangerous situation. So oxygen makes its way to the peripheral tissues and is offloaded from hemoglobin, and the little bit that's dissolved in the blood is also taken up by peripheral tissues. And then the cells release their carbon dioxide. So now carbon dioxide is actually a little bit mind-bendingly more sophisticated than oxygen transport. There's a lot of different ways carbon dioxide is able to make it to the exchange surface. And this is because carbon dioxide, oxygen is kind of a one-trick pony. The job of oxygen is to get to the cell to help oxidize glucose, right, to help with internal respiration. Carbon dioxide does a lot of other things. It's not just a byproduct. It acts as a buffer to the blood and helps with pH balance in the blood, etc. So it has a few more pathways that it involves itself in. First of all, some of it, like oxygen, just dissolves in the blood. Some of it's just going to go directly into solution. Some of it is going to be picked up by the red blood cells. And a portion of this will bind directly to hemoglobin. And hemoglobin will be the primary transport for carbon dioxide. Some of it, however, some of the carbon dioxide forms carbonic acid and dissociates into the conjugate base of carbonate and bicarbonate and hydrogen ion. The hydrogen ion is then picked up by hemoglobin. I've talked about how H+, or really the hydronium ion, is an effector of hemoglobin. And then the bicarbonate goes through what's called the chloride shift. It's exported via this antiporter. It's exported from the red blood cell, and we achieve charge neutrality by importing chloride. And this serves as an alkalizing buffer to the blood. It serves as an alkalizing buffer to the blood to counteract the effects of all this carbonate, this CO2, which can form carbonic acid in the blood. So this buffers the CO2 that's being dissolved in the blood. And then it travels to the exchange surface, and we do it all backwards. The chloride shift works in reverse. We import carbonate to the red blood cell and reconstitute carbon dioxide in the cell, which the portion that was being carried directly by hemoglobin is released as well. And all of this serves to exchange across the respiratory membrane. All right, you guys follow this a little bit? We'll talk a little bit about the chloride shift again when I get to the stomach and the production of HCl. There's something called the alkaline tide there that relates to this, but we're gonna save that for later. Okay, so pressure. Again, the scuba divers here know this, or any of, so for example, on the ski team this year, does anyone know Zane Fields? He went to, you know Zane? Yeah, he went to the NCAAs this year, and it was held out in Colorado, and, he is an exceptional skier. He really is an exceptional student as well. And he got 6th at the NCAAs, which is remarkable in and of itself. He probably would have been higher on that list, except the fact that he was several thousand feet above his training altitude. And his saturation dynamics in his blood were different than the people from the West Coast that he was training against. So as you move upward in elevation, your oxygen saturation dynamics goes down, and you can see here the highest established human civilization on Earth is around 5,000 meters, which is super high up there. If you think about that, that's 5,000 meters is 5 kilometers. That's a couple miles above. It's like 3.2 miles above sea level. Right? Jesus. It's really high. Those people, the few rare people that make it up to Mount Everest, they have very, very real problems with oxygen saturation at those elevations. All right. Last. Smoking. Okay. So on the panel on the left, you see respiratory performance. So this, there are different respiratory volumes. I didn't go through all of those for sake of time. I wasn't sure how much time I would have. And so I didn't go through all the respiratory volumes, but there are metrics for respiratory capacity. Your what's called vital capacity is the maximal amount of air your lungs can move. And that's one measure of respiratory performance. Your respiratory performance after about, I don't know, my age and after, I'm going to start towards the downward part of the slope here. Up until this point, it's pretty smooth sailing, but then everybody, as they get older, their lung capacity goes down. Right? It's just a normal part of aging. However, a regular smoker is on a much more rapid curve. So we see here this red curve, this rapid decline. And if you stop smoking, you are able to change the speed at which you lose respiratory capacity, and the slope of the curve matches or is parallel to the slope that you would find with normal aging. However, you never recover that lost capacity that you lost due to your smoking. And then you certainly can, if you are a consistent smoker, you can expect to have some sort of emphysema or respiratory disability, chronic occlusive pulmonary disease by the age of 65. And chances are extremely high that it will claim your life if you have smoked your entire life until the age of 70. So over 90% of lung cancer is due to cigarettes. This is a pretty high number. There are other causes of lung cancer. So, occupational carcinogen exposure. So, like, for example, mesothelioma, that is due to, for example, asbestos exposure, or a lot of other things you can breathe in that'll screw your lungs up. Around here in this part of the country, radon, one of the noble gases, a radioactive noble gas that is in high concentration in the bedrock, is a real problem in many homes because it's colorless, odorless. You don't know it's there. You can't tell it's there at all without a radon test. But it is radioactive and does cause lung cancer. So it is wise, if you are a homeowner or even a renter in New England, to inquire about radon levels in your basement. I don't know if you've heard of that before. Probably most of you have, I hope. Radon's real, and there are mitigations for that. And then, a little bit of air pollution. But the vast majority of lung cancer is due to cigarettes. So here is a dissection of someone who was a smoker. And you can see the black tar deposits in that tissue just from this. And down at the bottom, we have some resected lung tissue, happy pink on the left, which is hopefully everyone in the room, and the damaged lung tissue of a smoker on the right. So what does this mean? So my statistics are a little old. It is 2018 now, but the NIH in 2015, NIH estimates, I guess they're usually 2 years behind. So like in 2017, they estimated that 2015 lung cancer cost the United States $13.4 billion in direct care, in lung cancer care. And then a loss of productivity due to mortality from lung cancer morbidity led to an additional $36 billion. $36 billion. So altogether, this is about $50 billion. That's the complete value of all of the trade tariffs that our commander-in-chief has just proposed against the nation of China, the two biggest economies on earth. That's a huge amount of money. That's about $150 per person per year that you are directly spending. $150 of your productive output every year is being sucked up by lung cancer, the lung cancer problem here in this country. And lung cancer kills as many people a year as the next 4 cancers combined. So that's colon, breast, pancreatic, and prostate cancer. All of them are dwarfed by an entirely preventable, or at least a 90% preventable disease. 90% of this problem is because of cigarettes. about 250,000 new cases of lung cancer a year in the United States, 116,000 in men, 105,000 in women, and about 150,000 deaths. That's a lot of people. Men, the numbers for men peaked at around the mid-80s. Men have traditionally been much more, smoking has been more prevalent amongst men. However, with the women's lib movement of the 60s, part of that, an unfortunate consequence of that, was an increasing number of women became smokers. So that number has only in the recent decade begun to level off. As the market for smoking in men decreased, smokers were looking around to expand their markets, and one of them, an opportunity market, an opportunity population for smokers, was women. And so, sadly, women are afflicted almost as much as men today. Another population that has been disproportionately targeted by cigarette companies are African Americans. So here's a statistic. Black men are 20 to 30% more likely to develop lung cancer than white men. This is a population that is heavily marketed to. I grew up in inner city Detroit, literally surrounded by cool cigarette ads, and I find it to be disgusting. Is what I'm going to give you my take on that. I find it to be a often overlooked systemic bias that is unduly burdening this population. Everybody is a target of this problem, bears this burden, but this population certainly carries the burden. All right. I think that's it. So this may be the first lecture I've ever actually gotten through everything in a reasonable time period. Recap. I talked about respiratory anatomy, some ventilation, and then gas exchange. I spent about a full lecture divided between today and Wednesday on hemoglobin saturation dynamics, which is certainly the most challenging concept I talked about. I did go through some carbon dioxide transport, and then maybe a little bit soapboxy, but with the best of intentions, talked about lung cancer. Are there any questions?